One of the benefits of dealing with field of view and the diameter is how we can apply that to what we're actually looking at underneath the microscope. All right, so here I have in front of us three drawings of three fields of view, increasing in power. Okay, so the very first one that I have here is scanning power. So the scanning power, if we're looking through that objective, we calculated in the last video, so I'm just going to use those numbers, that the diameter from end to end, so remember that would be all the way across the entire field of view, was 6 millimeters. So in this view, in this circle of the field of view, is a very amazing drawing of cells that are underneath the microscope. So right now we're looking at a long chain of cells. So I did the same thing for our low power, where 10x, since remember with that calculation, or we had actually measured this in the last video, is 2.4 millimeters. And we have a chain of cells that are through the middle as well. And then a high power where our field of view diameter was zero point 0.6 millimeters. And again, I've got a couple of cells that are in here. So by knowing how big this field of view is, we can actually calculate approximately, again, approximately how big each one of these cells is, at least in width, if we're measuring it from end to end. So it's pretty simple in order to do this, right? So if we think about each one of these and their specific field of view diameters, we just have to compare the number of cells to that number. So the basic equation is you are going to take your field of view diameter and you are going to divide it by the number of cells that fit across that field of view. I just wrote a cruise because that's just something I do. So this is going to give you the size, again, approximately of the cell. So in this first example, we are looking at 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 cells that fit across this field of view. So our field of view diameter is 6 millimeters, and we're going to divide that by 12 cells. And that will give us 0.5 millimeters, which means each one of these cells that we see here, right, from end to end, is about 0.5 millimeters. Now, again, remember that we can convert that into micrometers if we need to, right, by multiplying that by 1,000. So we can convert that by multiplying this. I'm just going to do this total longhand here, which gives us 500 micrometers. So it's the same thing, right? We're just using different units. So we can, again, estimate about how big these are. Now we can do this for every single one. So if we then look at this next one, we have 1, 2, 3, 4, 5, 6 cells. So we can take our 2.4 millimeters and divide it by 6 cells. That will give us 0.4 millimeters. So again, this cell from end to end is approximately 0.4 millimeters. And then the last one that we're looking at over here under high power, right, is looking at 0.6 millimeters from end to end. And there are just two blobby things here. We'll call them cells. I don't know if they really look like cells, but whatever. My drawing abilities are amazing. So you take your field of view, again, 0.6 millimeters, divide that by 2, and that will give you 0.3 millimeters. So by knowing your field of view, you can estimate approximately how large each cell is in millimeters and in micrometers. And that gives you an idea of approximately how big these items are. So this doesn't just apply to cells, right? It can apply to basically anything you're looking at underneath the microscope. So you can then compare sizes. So for example, we were in a larger power, a smaller power here, right? We were only at using the 4x objective, which was a total magnification of 40x, right? So this is a 40x. And we saw cells here that look kind of small, right, in the drawings. And they're really 0.5 millimeter or large. We're just looking at them kind of from farther away. As we zoom in, right? So if we look at the last one, these two cells look gigantic, but they're actually smaller than the ones we were looking at over here. That's just because of the magnification that we are using. So we can then estimate that these cells are smaller than those based upon the magnification that we are using.